most people have been exposed to a coronavirus. Of course, coronaviruses in general are probably the second leading cause of colds in humans, but it's a different coronavirus. It's different than the one that's currently circulating, and so we don't have that underlying immunity to the one that causes the common cold.
Something to Chew On is a podcast devoted to the exploration and discussion of global food systems. It's produced by the Office of Research Development at Kansas State University. I'm Maureen Olevnik, Coordinator of Global Food Systems.
I'm Scott Tenona. I'm a philosopher of science.
And I'm John Fabian. I'm a food scientist.
Hello, everybody, and welcome back to Something to Chew On. There are times when we take stock of our lives, try to better understand where we fit in the natural world, and to find ways where nature manages to provide us with some answers. Today's podcast brings us a timely discussion with Dr. Megan Neiderwerder, Assistant Professor in the College of Veterinary Medicine here at Kansas State University. With the current worldwide coronavirus challenges, the topics discussed with Dr. Neiderwerder are not only timely, but instructional on helping us to understand what a virus is and how they impact the world around us. Megan's teachings cover viral diseases in swine, equine, avian, bovine, and canine species. Her research investigates control strategies for endemic and foreign animal diseases of swine. She is internationally recognized for her expertise in two primary areas of research, including the role of the gut microbiome on outcomes following viral respiratory infections, and the risk and mitigation of foreign animal disease introduction and transmission through feed and feed ingredients. Today's podcast is being recorded in a manner that I certainly never anticipated. John, Scott, Megan, and I are all in different locations. Courtney is recording via Zoom at her home. Due to the spread of the coronavirus, our new normal is to stay connected in an unconnected environment. We've talked frequently about recording podcasts with someone that was off-site, but I always figured that John and Scott would be in the same room with me chatting with someone else remotely. The time that we are in today is quite different, and the coronavirus spreads and keeps social distancing as part of the new normal. We are each in our own space and hoping for science to help bring a return to a healthy world. With that, I would like to welcome all, and specifically welcome Megan to the podcast. The podcast in this series is focused on the food system at Kansas State University, and certainly animals are a critical part of that system. This is actually the 20th recording in this series, and I was surprised to look back at the list and realize that this is the first podcast that we've done on animal-related research. Between the College of Veterinary Medicine, the College of Agriculture, and the Departments of Animal Science and Grain Science and Industry, there's a substantial amount of work being done in this area, whether it's through animal health or food and feed, and just a whole bunch of other areas that we touch on in the area of animal health. So, Megan, I've got a ton of questions for you as we go through this. I doubt that we'll get to all of them, but what I'd like to start off with is getting to know a bit about you and understanding how you got interested in this area, and then we can see where the discussion takes us. So, Megan, could you tell us a little bit about yourself?
Sure. Well, thanks to everyone for the opportunity and invitation to speak with you, and thank you for your work on the global food systems. I think it's really important, and like you said, important to talk about animal health and animal diseases as part of the efforts for global food systems. So my name is Megan Neiderwerder, and I am a veterinarian. I actually went to Kansas State for veterinary school. I've been a veterinarian for about 10 years now, and as part of my veterinary practice, I worked with dogs and cats, zoo animals, as well as pigs. And when I decided to come back and pursue a career in academic research, it was really based on my desire to provide information or to research questions with regards to population health and livestock. And I've always had an interest in infectious diseases and the ability to ask questions. How do things work? How can we improve animal health? And the opportunities that research affords in an academic setting is to really understand how we can improve animal health at a population level. And even in my case, it's my research is focused on swine health. How do we improve the health of pigs around the world?
How long have you been on the faculty?
I've been on faculty now for almost 5 years at the College of Veterinary Medicine in the Diagnostic Medicine Pathobiology Department.
Great.
Could you tell us a little bit about what your research is on swine health?
Sure. So I have really two main areas of research, and both again focused on viral diseases in swine. One of the areas that I have focused my research on is looking at the utilization of the gut microbiome to improve pig health in the face of respiratory disease. So when we think about pigs after they are weaned, pigs are typically weaned about 3 weeks of age from their mothers, and they have a high incidence and high susceptibility to respiratory disease at that time. So we typically call that porcine respiratory disease complex, because these respiratory diseases often have a viral component, a bacterial component, and often are due to environmental stressors as well. And so some of the respiratory diseases that are currently challenging pigs throughout the world have been around for many decades. And so that means, we're at a place where we want to look at alternative approaches for how do we improve the health of these pigs in the face of respiratory disease with alternative strategies. And so the focus of my work has been, how do we utilize the gut microbes and the beneficial bacteria in the gut to actually improve or enhance animal health in the face of the respiratory challenge environment. So again, focused on the main virus that I have on is porcine reproductive and respiratory disease syndrome. It's really considered the most costly disease of swine production in the United States currently. And my work is really focused again on those gut microbes and how do we improve disease. And the second area of research that I have is looking at foreign animal diseases and the risk of foreign animal disease entry into the United States through feed and feed ingredients. And so that work has been focused on the fact that we import millions of feed ingredients, millions of kilograms of feed ingredients from all over the world that then are used to feed and provide nutrition for our pigs in the U.S. And so with the introduction of porcine epidemic diarrhea virus, we started to recognize that feed ingredients may be a potential route for transboundary animal disease spread. And so my research has really focused on understanding the risk of other foreign animal diseases, such as African swine fever virus in feed.
Cool. I was
interesting. Yeah.
So I was going to ask just about the first set of things. So how is it that the gut microbiome for pigs actually affects their health? Does it prevent them from getting things? Or are you talking about recovery time? Like, what's the relationship there? And it's curious because that's not something we totally normally think of, right? So this is the gut, and you're talking about respiratory disease, right?
Yes. It's not an inherent thought to think about our diet and the microbes in our gut affecting our health outside of the gut. It's more inherent to think about how those beneficial microbes may help resolve diarrheal disease. But in fact, they really play a role in systemic immunity throughout the whole body. And so what we think happens is that the gut is really the largest immune organ in our bodies. And so the way that those gut microbes then communicate with the mucosal immune system in the lining of the gut impacts how the immunity responds, or the immune cells respond to pathogens in other areas of the body, such as the lungs. So when we think about some of those beneficial microbes, not only stimulate the immune system, but they also communicate with the immune system in different areas of the body through metabolic products of those microbes. And so we think there's a couple of different ways. Oftentimes the exact mechanism by which these gut microbes impact disease outside of the gut is not fully elucidated. But what we see is that pigs respond better to a respiratory challenge. So let's say they have fewer lung lesions, or they gain weight normally, or they replicate the virus to lower levels. So it almost creates a more resilient or resistant pig when they are exposed to these pathogens.
Oh, that's fascinating. I'm fortunate to be blissfully ignorant on most of this area. Is that confined? Is that phenomenon confined to monogastrics, or does it extrapolate to ruminants?
Well, we could assume that it extrapolates to ruminants, but this is really a new area of research. So a lot of the research that's been done has been done in mice models of respiratory disease. There's a great study with pneumococcal pneumonia and a few other respiratory disease pathogens where they will see that the presence of endogenous microbes or fecal microbial transplantation, or maybe those mice that did not receive antimicrobials and have more diverse microbes, actually respond in a better or more robust way when they're infected with respiratory pathogens. And so a lot of this research is fairly new, but when people ask about what impacts the microbiome or what the microbiome impacts outside of the gut, it's sort of easy to go, well, probably most things. We're just learning, and we're just at the cusp of learning exactly what those are.
Wow, thanks.
Yeah, crazy new world of science here for all of that, right? Are there particular strains? You said something about the diversity of the gut microbiome is important, or what kinds of things in particular are you looking at, right? So there are particular microbes that you're investigating, or your own work? Are you looking at the diversity? Tell us a little bit more about what's going on there.
Sure. Yeah. So one of the most interesting things about some of these respiratory diseases in mice models, but also in pigs, is that a fairly common and consistent finding is that the more diverse the microbes are in the gut, the better the animal responds to respiratory disease challenge. So, and that's been shown in some of the work that I've done with pigs, both, let's say, if the microbiome is more diverse prior to challenge, almost like there could be a prophylactic or preventative measure with regards to the microbiome on subsequent challenge, which is somewhat exciting because it gives you the idea that microbiome modulation may be able to prevent or at least put a pig in a better place to respond to disease prophylactically. But some of the microbes that we found that specifically seem to be beneficial, we found that a non-pathogenic E. coli seems to be beneficial in the pig gut. We found that increase in ruminococcus seems to be beneficial when it's detected in the gut. And then also fecal microbial transplantation. So we've used fecal microbial transplants from older aged sows that had high histories of health and great litter characteristics, took the feces from those sows, transplanted it into younger pigs, and actually showed that fecal microbial transplant can provide benefits with regards to respiratory disease when given prior to challenge.
Wow, that's fascinating. Absolutely fascinating. And in the things that we're looking at, I hesitate even making this jump at this point in time, but in the things that we're looking at going on in the world today with coronavirus and these viruses that are in humans, what are the chances that the kinds of things you're looking at now in swine and in animals is the same as what you're going to be seeing in humans in helping to manage some of these things?
Yeah, I think some of the similar parallels that we've seen is when we talk about foreign animal disease, such as porcine epidemic diarrhea virus, when that was introduced into the U.S. swine herd in 2013, within 8 weeks of its first identification in a pig herd, it had been detected in almost all of the major swine producing states.
Wow.
And the reason that is because we have approximately 1 million pigs on the road every day being transported between different farms. And everything is fast paced. Pigs are moving. we see this, even within the country, let alone global distribution. We see that, the movement of people and animals happens very quickly. And so we've seen that with the coronavirus as well, in that the global movement of people makes it difficult to contain these viruses once they're introduced. The other sort of interesting parallels when we talk about African swine fever virus, the USDA released, I think 2 weeks ago, that if African swine fever virus is introduced into the United States, which of course we're doing everything to prevent that, there will be a 72-hour standstill where no pigs will be moved. And so again, as you think about that in parallel to what we're all currently doing, which is trying to stay in our homes and not be exposed to other humans, there are some similarities, and that just goes back to how these infectious diseases can be spread because of how rapidly humans and animals are moving throughout the world.
Mm-hmm. So even in cases, almost as though humans are a secondary vector moving the pigs around.
Well, so pig production in the U.S. is fairly specialized. So when you think about the different age groups of pigs, there are certain farms that specialize in pregnant sows or gilts and farrowing, so that they have young piglets that they nurse. And then those piglets may be moved to a nursery facility where they live for several weeks prior to going to a grow finish facility. And then those pigs may be moved once they're ready to go to market. And so this has increased the efficiency by which we can produce pork in the U.S. Because of these specialized operations. But it also means that pigs are frequently moved, during production.
Sure.
Do you have an opinion on that trade-off, right, between the efficiency and basically the security of the food system there to an outside pathogen, to a foreign pathogen?
Yeah, I think that the movement of pigs can sort of unveil the vulnerability of a pathogen being moved. But I think the focus, of course, one is prevention of any pathogen entering. Mm-hmm. The second is rapid identification of pathogens if they do become introduced, so that if a pathogen is introduced, there are protocols and procedures in place to stop pig movement so that, again, the pathogen can be contained and eradicated as quickly as possible. So, I think as these diseases are introduced, similarly, when I talk about PEDV, it changes people's mindset and, again, can help us learn about the best way to prevent the spread of any virus or bacteria if it's introduced.
We sort of moved off in one direction, and you started to talk a bit about, I think it was feed materials or feed ingredients for the animals as a source. What ingredients are we particularly concerned with?
Sure. So some of the research that we've done has looked at what's the stability of foreign animal diseases in feed and feed ingredients. Mm-hmm. Can viruses be transmitted through the natural consumption of plant-based feed? Mm-hmm. And then how do we mitigate the risk of those viruses in feed? And so the first part of this research was really to understand if we inoculate feed ingredients and subject those feed ingredients to temperature and humidity conditions that simulate transoceanic shipment, because this is what we're concerned about. If feed ingredients are being imported from China or from other areas of the world that have different diseases or foreign animal diseases, could the virus survive and would it be stable? And so we've looked at various ingredients such as organic soybean meal, conventional soybean meal, pork sausage casing, some pet foods. And what we found is that it looks like soybean meal supports the stability of viruses such as African swine fever virus, classical swine fever virus, and pseudorabies virus. And so it looks like there are certain ingredients, and I think it has to do with the protein content of these feed ingredients or the moisture content, but there are certain characteristics of individual feed ingredients that promote stability of these viruses. And of course, those are what we're concerning high-risk ingredients for import.
Right. That's truly something that a lot of people wouldn't even think of.
No.
when we think classically of viruses, we think that they need some kind of host to survive unless they're very, very dried down or low water activity. And
Yes.
this really opens the window on something that needs to get to be figured out and at least approaches to controlling it.
Yeah, and I think that goes back to understanding sort of the global distribution of many commodities, and that includes feed ingredients for our livestock here in the U.S. So certain feed ingredients that we put into the feed, some of which we do not manufacture in the U.S., we can only get from China. But there are other feed ingredients that we could locally source that, of course, would reduce the risk with regards to introduction of viruses onto the farm.
Right.
Interesting. The comment of only being able to source materials internationally is very true in human food consumption as well. I recall some instances in the past where there were challenges in buying from certain areas, and we realized in the food industry overall that some of those were the only places you could buy those particular ingredients. And just the idea that those viruses can survive on some of these things is a little scary. And as John said, it's going to be something that we really need to better understand the how and why and how to control.
Yeah, I think we've operated with a lot of assumptions that were untested, and that the time now has come to really test those and get solutions. Can
we take this back probably as much for our listeners as anything to the basics of what a virus is? Can you talk a little bit about what a virus is and what makes it so special? What makes it different from other organisms in the characteristics that it imparts here?
Sure. So a virus, one of the biggest differentials from other organisms that a virus requires is that to replicate a virus requires a host cell. And so when we talk about replication of virus, let's say in a pig, for a pig virus, it requires the pig to actually replicate the virus particles so that there is more virus. There's an important point there when we talk about contamination, because the virus is not replicating when there's no host cells. But if it's a highly stable virus, that's when we can see the virus essentially being maintained as infectious. There's a couple of different characteristics about viruses that I think are important to always think about. One is that viruses can either be made of, the genetic material can be either RNA or DNA, and it can be single-stranded or double-stranded. And then another characteristic of viruses that we typically talk about is that viruses can either be enveloped or have a lipid membrane, or they can be non-enveloped. And so just general characteristics of viruses, if we talk about a non-enveloped virus, it's typically much more stable in the environment. It's less sensitive to disinfectants. And then when we talk about an enveloped virus, typically those viruses are more sensitive to disinfectants and less stable in the environment. And the other factor that we think about is when we talk about DNA versus RNA viruses typically have higher mutation rates because they have less proofreading when they replicate themselves. And so when we talk about RNA viruses such as influenza, and of course we know how much influenza changes every year. This is one of the reasons we have to get a new vaccine each flu season. When we compare that to, let's say, a DNA virus, DNA viruses are typically much more stable, less mutations. So there's certain things about different viruses. One of the characteristics of African swine fever virus is that it is a very large virus. So some viruses are, maybe 10,000 bases. We talk about African swine fever virus, it's much larger, much more complex. And when it has much more complexity with regards to the genome, that means it produces more proteins. And that's when we see challenges with regards to vaccine production. So one of the biggest challenges right now with African swine fever that I'm sure you've seen in the news is that there is no currently commercial vaccine that we can use to protect our pigs.
Right. in essence, the viruses take over or they hijack the host cell's enzyme and other systems to make more of themselves, and that's something that right. So what are the envelopes made of? Are those polymeric sugars, or I would have expected them to be more resistant to cleaning solutions, for example.
Yeah, that was my thought as you were describing this.
Yeah, when we think about the envelope, it's mostly lipids and then has proteins on it. Viruses typically use proteins on the surface, glycoproteins on the surface, to actually bind to the host cell receptor, enter into the host cell, and then replicate. But when it has that envelope, it often has those lipids that make it more susceptible. When we talk about non-enveloped, it's proteins that are surrounding the genetic information, and so it's more stable.
I was just going to say, so is that one of the reasons soap's particularly effective? Is that like really just done on the envelope viruses, you know, attaching to the lipids on the protein, or do they work? Is soap as effective in non-envelope viruses?
Well, it depends on the virus, of course, but in general, a lot of those characteristics, of course, when we talk about hand washing and the importance of hand washing, a lot of it is the mechanical, cleaning of the hands, but of course it depends on the different viruses. But yes, in general, these soaps are fairly effective against a broad range of viruses, but it again sort of depends on the virus. Some viruses are very hardy and very difficult to eliminate from the environment.
So this is random, but here's a question: are viruses alive in your opinion? I mean, because they're their own little strand. They can't replicate on their own, right? When I was a kid, I was taught one of the things of one of the properties of life was that it, is replication, right? And I know at the time people disputed whether viruses are alive, right? Because they can't replicate themselves; they need a host. Do you have an opinion? Do you care?
That's sort of like the day one virology question for the students.
Cool.
So, would you consider it alive or not alive? And it's interesting because when you actually talk about it in, when we talk about diagnostics and what we think about with viruses, we more use the terminology infectious versus non-infectious. So because one of the biggest diagnostics, one of the most widely used diagnostic tests to detect viruses is PCR, polymerase chain reaction. Right. But that test looks for viral genetic material. So it would look for the DNA or the RNA associated with the virus. But if you have a positive PCR test, it doesn't necessarily mean that virus is infectious. So those are sort of the terminology. When we think about infectious viruses, we think about, can that virus, if it was exposed to a susceptible cell, would it be able to gain entry into the cell and replicate and be infectious to that cell? So really, those are sort of the terminology, at least in diagnostics, that we think about. But the whole is a virus alive or not alive? I think people have different opinions, I suppose, on that, which maybe makes it, again, a good academic question for the students.
Right. So here's just a random question then. In terms of terminology for disabling a virus, what's most often used, right? Sort of kill would, right? People say they kill the virus.
We usually say inactivation of the virus, so that, because what could happen is, let's say, and we've done a lot of these experiments in feed, and let's say we expose the feed to a heat treatment or some sort of chemical that inactivates the virus. If we take that feed and test it on PCR, it may still have viral genome, but if we test it on cells, it wouldn't be able to infect the cell. Maybe that's because the lipid envelope has been disrupted. Maybe that's because the glycoproteins now can't bind to the host cell. There could be some different factors, but what we would talk about that is inactivation of the virus.
One of the areas of study that you had listed there is control strategies for endemic and foreign animal diseases. Can you speak to what some of those control strategies are, or things maybe that you're researching?
Sure. So when we think about in general, diseases in livestock, we first, of course, would like to eradicate or eliminate the disease from a herd. So we think about PRRS virus, ideal scenario is that we would eliminate that from a herd. But then if you can't eliminate it, or if the herd is endemic for that disease, then you start thinking about, can we use a vaccine strategy to improve the response of pigs to the virus, which there are some vaccines available for PRRS. And then sort of third or are there alternative strategies to actually improve the response of the pig to the virus when it's exposed? And so some of the strategies that have been looked at in that area is, are there certain genetic properties or genetic aspects of the pig that actually improve its ability to respond to viruses? Are there microbiome characteristics of the pig that improve its ability to respond to viruses? And so the focus of my work in that area goes back to that microbiome. Can we sort of set up the pig's gut health to help the pig respond to any pathogen? It's really the goal, but, one of the largest sort of economic losses in pig production is these respiratory disease susceptibility of those young pigs, 3 to 5 weeks of age, right after weaning. So we think about, how do we improve gut health to improve respiratory health, and even as sort of a supplement to all of the other activities that are going on, how do we reduce the stress of the pig so that it can respond better to infection? How do we improve the nutrition of the pig so it can grow normally even under disease challenge? So we kind of think about some of those things. With regards to foreign animal disease, we really think about, how would the virus be introduced into our country and how do we mitigate that risk? So some of the big aspects of foreign animal diseases, particularly with African swine fever, that we consider is contaminated pork products. We know that the virus survives for long periods in pork, and so that can be a risk factor for infection in pigs. And then, of course, we talked about feed and feed ingredients being a risk factor, and understanding how we mitigate that risk for disease introduction.
So when you say pork, does that mean that humans are vector transferring the diseases back to live pigs in the U.S. possibly, or how would the transfer happen from imported pork?
So importantly, African swine fever virus, humans are not susceptible to that virus. And pork is still safe to consume even if pigs are infected. So that's an important point to highlight. When we talk about pork products, and what we've seen in other areas of the world is that oftentimes people, may bring a sausage in with them through the airport and not think about the risks that they are bringing in potentially contaminated pork products into the U.S. It's illegal, and but there have been confiscations with regards to finding these pork products that should not be brought into the United States. And one of the big aspects with regards to foreign animal diseases is educating the public so that they understand that bringing in products from other countries into the U.S. can be a huge risk factor for our animals here as well as our crops here.
Fascinating. Explain the impact on crops. Or are you just saying if they brought some other crop-carrying disease into the U.S.?
Yes, crop-carrying disease. So when you think about you go through the airport and they ask if you have an apple or a banana or any meat products, the biggest reason for that is that there are certain agricultural diseases, pests that we do not have in the United States that we want to maintain our crops, our animals free from. And so when people bring in agricultural products illegally from other countries, the risk is that those commodities, those products could potentially have diseases that we do not have here.
Sure.
That was one of the fundamental differences in approach between scientists that wanted to go around the world finding new varieties of various and sundry crops versus people who wanted to keep them out via inspection. And actually, the son of one of the first presidents of Kansas State was one of the folks that was critical in going around the world and bringing in new varieties of agricultural products
and shipping them over.
Yeah. I think there's definitely a balance between, learning from different agricultural products that could be beneficial, and then making sure that those agricultural products don't contain microbial pathogens that would, impact the rest of the crop. So,
yep.
Do we find that the contaminated materials are more frequently sourced in maybe developing parts of the world? Are there differences in different areas around the world that cause specific problems in products that are moved into the United States, or is this just it kind of is different in different areas around the world, and you just have to be careful regardless of where you're shipping from?
Yeah, I think you have to be careful regardless, because what we've seen oftentimes is that maybe a product is manufactured in one country and maybe it's processed in another country, and then maybe it's shipped to a secondary country before. So there's all these steps, and again, that goes back to efficiency of the global trade that has been established over years to make these products available to people all over the world. But there's risks with regards to determining where the product came from, and if there was any risk upon its manufacturing or processing that puts it at risk for contamination of the diseases that are in that country. We know, for instance, that there are crops that are grains and crops that are dried on the roadways in China. And those roadways are, of course, shared with trucks and vehicles that could be transporting pigs. And so when you think about that and the potential for those crops to become contaminated because of these sort of risky agricultural practices with regards to drying on the roadways, you can imagine how those feed ingredients may become contaminated in the environment and then could potentially maintain the virus during shipment. So it sort of depends on the ingredient, but it depends on where it's coming from too.
Sure, that makes total sense.
Yeah, I've seen that in India as well.
Could I ask a question back to gut microbiome? Are there any standard practices that you'd recommend of farmers to be putting into place to improve the health of their herds, or are there particular kinds of things that you would have to do, like the fecal transplants? So are there standard things that we do that actually that hurt our swine herds, or are there particular that would make it really easy to improve their gut microbiomes, right? Or is this something that would be actually kind of complicated to do or would require new techniques?
Well, we're really still learning about how something like fecal microbial transplantation or microbiome modulation could be implemented in a broad scale in swine herds. But one of the biggest challenges with regards to fecal transplants, even on the human side, when we talk about, so in humans, fecal microbial transplants are most often used for recurrent Clostridium difficile infections that have been already treated with antimicrobials. And so this is somebody who's been treated with antibiotics probably multiple times and then gets the fecal transplant, and oftentimes it has a fairly high success rate. When we talk about that in humans and even in pigs and some of the work that I've done, we don't have sort of the foundational knowledge to say, let's take feces from 10 pigs or 10 humans, and based on the microbial characterization within that fecal transplant, I can pick out the ideal donor. We don't really have that information. So when you think about who is going to be your donor for fecal transplants, oftentimes you go off of phenotype. So let's say the pig grew really well. The pig has had a great health history. The pig doesn't have certain pathogens circulating. It's very similar in humans. You talk to the donors to say, have you had any, history of diarrhea? Have you had, these infectious diseases we need to screen you for? But so you're sort of looking at the phenotype of the person as opposed to, or this pig, as opposed to the genetic characterization or the microbial characterization of the actual transplant. And I think once we get there, then we'll be able to really sort of implement this more on a broad scale, where we go, these are the characteristics of a transplant that really provide benefits under these circumstances, or in our modern day pig production.
That's
and like you have a job in front of you. Exciting times. What's the Chinese proverb or the Chinese curse? You should live in interesting times.
Yeah, we're in that. Yeah. What were you going to say there, Scott?
Oh, I was going to ask more about the gut microbiome, but I've got another question that's been floating around in the back of my head. And Megan, I was wondering if you could say something about what makes a virus easily transferable, right? So one of the things with the novel coronavirus, right? So this came from a reservoir. And my understanding is that I think the reservoir was bats, right? And that it was living in bats and then transferred through some other animal and then finally got to humans, right? And so there's something about these kinds of viruses that once they leave that reservoir that animal reservoir where they were hanging out for a while, maybe without actually hurting that population, but they transfer to humans and they're particularly, problematic for us. This is a bad one right now. What makes a virus, first of all, easily transferable like that? And what about that kind of thing makes it dangerous in a way that some of the viruses that you're studying in swine are, there's no risk of transfer or infecting humans in a way that's going to hurt us?
Yeah, I think one of the important sort of components to what's going on with the coronavirus is that, as you said, the way that the humans were exposed to it was this very close interaction between humans and wildlife, where we're seeing that maybe a virus that has been around in the wildlife population for decades and has never caused a problem to that wildlife species or to humans because of the lack of close interaction. When a human is then exposed to that virus, can become infected. And I think one of the important things about viruses is that they have this high ability to mutate and to change. So, of course, most people have been exposed to a coronavirus. Of course, coronaviruses in general are probably the second leading cause of colds in humans. Mm-hmm. But it's a different coronavirus. It's different than the one that's currently circulating, and so we don't have that underlying immunity to the one that causes the common cold. And so, one of the things that can make viruses transmissible is, of course, that oftentimes viruses are transmitted through the oral nasal route. If you think about respiratory diseases, those are ones that can make you cough, can make you sneeze. That can create these aerosols that, again, oftentimes can contain large amounts of virus if a person is in the acute phase of infection. The other thing about viruses is that even though they require cells to replicate, some of them can survive in the environment for an extended period. And again, that goes back to, DNA versus RNA, envelope versus non-envelope. But when we think about touching handle doors and touching our phones and those kinds of things, we want to be sure that, we're taking the precautions to not only wash our hands, but think about the types of materials that our hands touch throughout the day and make sure that we have those clean. So those are a couple of the characteristics with regards to viruses.
Thanks.
And is there no, I was just going to say, so is there anything particular about the type of virus or what it is that makes it particularly harmful to humans, right? Sort of so why is it that the viruses that you're studying that are cause a lot of disease in pigs, right? Sort of don't happen to cause diseases in humans. Is it just kind of, luck, or is there some particular characteristic about them that makes them not dangerous to humans, but something like the novel coronavirus, right? Transferable, and it's going to infect us and make us sick.
Yeah, typically the way that the virus is determines what host to that is susceptible is based on the proteins on the surface of the virus. So those proteins only recognize certain host cells. And then also on the host side, the host has to have the correct receptor, or it's often a protein again on the host cell. So those interactions are what allow the virus to actually take its, have the receptor bind to the protein bind to the host cell receptor and enter into the cell and cause infection. But viruses have a wide range of how they cause disease. When you think about a virus like HIV, people have HIV as lifelong infections because that virus actually integrates itself into the host genome. Whereas you think about other viruses and the infection is fairly short. Maybe it's 3 to 5 days, and that's the only time period when you are shedding the virus, and then your immune system clears the virus. Some viruses cause chronic infections. Some are very acute. Some cause, like African swine fever virus in pigs, causes extremely high mortality. So we see almost 100% mortality, whereas other viruses have mortality rates less than 1%. So really depends on the virus and the mechanisms by which that virus has evolved to cause disease, and also sort of this variation that we can see with regards to the length of time that the virus may infect the host. And again, host susceptibility has a lot to do with the actual cells of the host and then the proteins on the virus.
Interesting. Thanks.
Well, that's very interesting. What I was going to ask, we're going to be putting on a workshop. Global Food Systems is going to be putting on a workshop on microbiome in another month or two, and hopefully it'll be face to face. But if we're still in the situation we're in right now, we may try and take it to a virtual level. But on the microbiome, as I was putting the ideas together on this, and Megan, thank you for your help in working with me on it. There are a whole lot of different areas working on microbiome, and I wonder in my mind how those areas overlap, or if they do not overlap. What are the similarities and differences between, say, soil microbiome and what you're working on with swine gut, or things that are being worked on in poultry gut? Or is that a fair question to ask you? I mean, it may be that the study areas are different enough.
No, I think it's a good comment to have about the microbiome space, because when we think about the research, really it's been over the last decade that we've seen this expansion in microbiome work, and really the vast majority, every year the number of microbiome publications increases on both the human side and, let's say, the swine side. There's probably over 50,000 publications now on the human side, but the vast majority of those have been in the last 3 to 5 years. And even on the swine side, we talk about maybe 1,000 publications on the swine side. So it's this new area of study that is very complex. And when we think about the microbial communities in soil, or in our guts, or in the guts of pigs or poultry, and we think about all the interactions of those microbes, it's very complicated. It's very difficult to replicate, let's say, outside of the host, because you think about the immune cells of the host impact the microbial replication in the gut, and then the microbes within the gut interact, and the metabolic products impact what other microbes may do or may produce. So I think there's a couple of challenges with microbiome research that is probably fairly consistent in any microbiome research, one of which is replicating what happens in the natural environment in a laboratory setting, but also how you actually analyze the microbiome. There's many different areas. You can use metagenomics, you can use whole genome sequencing, you can use 16S ribosomal sequencing, which is based on bacteria. You can use microarrays to look at populations of microbes. You can look at transcriptomes, so maybe what the microbes produce, what sort of the response between the host and the microbes. So I think it's such an exciting space because we have so much to learn and there's so many different areas. And really, if you look and see, an aspect of the microbiome, again, more than likely, it probably has an impact on some sort of disease in the host or in the, in maintaining a healthy crop in the soil. And so I think it's really exciting, but again, I think it's important for people to sort of wrap their head around the complexity of how we research the microbiome.
True. Another term I've heard used in that reference is the microbial ecology of the gut. Is that a
Yes.
correct? Okay, good.
Yes.
Maureen?
Well, I was just going to say, with what little I've probably read and understood enough on the microbiome to be really dangerous in the area, but what I have read has absolutely fascinated me, and it seems, from my limited understanding, that it's got the potential of changing the way we look at health in many ways, the way we are looking and understanding what's going on in our systems and how to manage and control healthful activities. Is that a fair assumption on my part?
Absolutely. I think one of the most sort of fascinating and exciting things that I think about is, in the future, are we going to be inoculating our young babies and, on my side, the young pigs with microbes that we know will improve the health of that person or that animal throughout their lifetime? That's really exciting because you go, wow, we can, if we learn about these microbes, what they do, and then on the human side or on the pig side, understand what diseases they're going to be susceptible to later on in life, and give them those microbes that are going to help them be resistant or less susceptible to those diseases. That's a whole nother avenue to think about how we can prophylactically or preventatively provide health to people or pigs, prior to or early on in life. I think it's really, really fascinating and exciting. And we think about we are what we eat. We haven't really talked about the diet, but the other fascinating thing about the gut is that everything we eat impacts the microbes. So when we talk about learning about the connection between nutrition and microbes and overall health and neurologic conditions that maybe even come in later on in life, it's really, like you said, I think fascinating and exciting. I think, it'll be very, very cool to see where we're at even, 5 to 10 years from now in the microbiome world.
Absolutely.
Absolutely.
Yeah,
what comes to mind with what you had just stated was just some reading I did it's been a while ago, but on childbirth and on the difference in the potential of the child with a vaginal birth as opposed to a cesarean section and the number of microbes that child is introduced to during that process. And just thinking about all of those kinds of things is, as I said, fascinating. I mean, it makes your mind kind of swim on all of the areas that those microorganisms are going to be touching and relating to how well we are in the long run.
Yeah. Well, and in nature, there's at least one example in nature for that. Koalas are born without, if they were, there's a transfer of microbiome from the mother to the baby that allows them to eat and digest the eucalyptus trees, which they wouldn't be able to otherwise. So
interesting.
There are examples in nature of what we're trying to do.
Yeah. So has the feed of swine been studied in terms of the effects on the microbiome and their health, right? So how much do we know about like what the diversity or the type of feed that pigs get affects their gut and that affects their health?
Yeah, so a lot of the focus, of course, in the nutrition side is what components of the pig's diet do we need to make the pig have the pig gain as much weight and as quickly as possible and maintaining health. I think an area there that we should sort of bridge the gap is as we understand the best nutritional outcomes with regards to growth, also thinking about are those the best nutritional aspects if that pig is in certain health challenged conditions. And so oftentimes we do these the research studies in herds that are fairly healthy that maybe don't have a disease challenge. And then when we get into a disease challenge situation, we understand that pigs not only eat differently. Of course, if a pig is not feeling well, they may not eat as much, but they also metabolize and take nutrients from the feed in different ways based on their health challenge. And so I think that's another sort of exciting area where we can maybe bridge the gap in different disease settings. What sort of nutritional aspects of the feed should we shift to, again, set the pig up for the best possible health outcome?
And does diversity of feed correspond to diversity of microbiome?
Ooh, off the top of my head, I don't know if we know that. If we think about, you mean like if a pig was fed different diets throughout its lifetime, would it increase the diversity of the microbes?
Right.
Yeah, I think that's an interesting point because you think about the feed source, of course, in most high biosecurity operations is highly standardized and changes as the pig ages. And so most pigs are not fed, different diets or, a wide range of different diets. As you think about maybe in developing worlds, when people hold pigs in their backyard, we talk about backyard operations. We talk about this with regards to disease. Oftentimes those families may feed the pigs scraps from their meals. And so, of course, I don't know if there's been a good study looking at, if a pig is exposed to a wide range of food throughout its life, what happens to the microbes. But my assumption would be, yes, that the diversity would increase.
Because a lot of the work to this point has been more directed phenotypically. You're trying to change some entire animal characteristic and improve it or keep it the same, as opposed to going at the genotypic level.
I think this has been a great conversation. Obviously, very timely. I've learned a lot. It's been great.
Truly.
Oh my goodness!
Thank you ever so
fantastic
much.
Work.
Yes, thank you for the discussion. It's been, I've really enjoyed talking with you all, and hopefully we can all meet face to face soon. But yeah, thanks again for the opportunity.
You bet.
Well, we'll look forward to the face-to-face, and thank you all for coming online. And I learned a ton here today.
Likewise. Thanks again.
If you have any questions or comments you would like to share, check out our website at ksu.edu/research/global food and drop us an email. Our music was adapted from Wayne Gowen's album, Chronicles of Carmela. Special thanks to him for providing that to us. Something to Chew On is produced by the Office of Research Development at Kansas State University.